So what Ed asked me to do was to sort of review where we are with low doses, adaptive responses, and bystander effects. So what I thought I'd better do is at least introduce the issues in this field because I don't think there are very many radiation people here. So I was going to do a little background on low dose effects, then talk about some of our new bystander data, which is suggesting that UVA is at least a signal, maybe not the signal. And then I want to go through some of our old and new low dose chronic data to address some of the questions that are really important in radiation protection at the moment, such as how do you relate chronic exposure to acute exposure? How do you relate alpha radiation effects to gamma radiation effects? And something that we've become very concerned about recently is how valid are the in vitro models that we've been using for years and years in radiobiology when you do the same experiments in vivo. How valid do we find the in vitro models? So my background to non-targeted or low-dose radiation effects. Most low-dose effects are non-targeted effects. And non-targeted effects include bystander effects, which are where you get effects in cells, organs, organisms, which have not been exposed directly. So there's been no dose deposition, which is how radiobiologists think about irradiation. They think about the dose and the energy being deposited in a cell. But in the bystander effect, you don't need that energy to be directly deposited. And what you get is secondary effects due to signaling or cell communication. And there's a lot of interest in what these do to radiation protection. Do they increase the risk or do they decrease the risk? They've been known in the literature in cell culture. They were only discovered around 1990. But in the old literature, clinicians were well aware of so-called abscopal effects. They can be traced back to 1905. And this is where you irradiated an organ, usually with a tumor, and you get shrinkage of metastatic deposits elsewhere in the body. And then another phenomenon that's sort of related is so-called plastogenic factors. And these are factors that circulate in the plasma of people who have had radiotherapy or A-bomb survivors, these kind of cohorts. And they can cause chromosomal damage in if you transfer the serum to lymphocytes in culture. You can get chromosomal damage. So again, it's an indirect effect of radiation. And then Colin and I, with our group, did experiments where we would irradiate fish and put them swimming with unirradiated fish. And we were able to get effects in the unirradiated bystander fish that were almost identical to the effects in the directly irradiated fish. So that was a fish-to-fish communication. And then the other kind of non-targeted effect is genomic instability. And this is where you get effects in unirradiated progeny of the progenitor cells. So you irradiate cells, you let them go through multiple generations, and then you get de novo expression of mutations that were not introduced in the progenitor cell by the direct irradiation. So you've got a destabilization of the genome. And these are causing a lot of angst, really, in radiation protection circles, because we don't know how to handle them. We don't know what they do to the risk, which, as Jerry very clearly told everyone, is at the moment extrapolated from high doses. We assume that you just get less of the same effect as you decrease the dose. So the issues, they're pretty well what I've said. What do low doses do? What is the shape of the dose response curve, if you want to put it in physicsy talk? What do chronic exposures do? People are very conserved about the dose and dose rate effectiveness factors for low doses because they need to relate chronic exposures to acute exposures. A big question in protection is what is the low dose alpha RBE? The literature suggests that the RBE, or relative biological effectiveness, for alphas is about 10 times the equivalent dose of gammas or X-rays. But in the low dose region, nobody knows. We have no idea. And I'm going to show you some data that suggests it's nothing like 10, 20 or 40. Do alpha RBE values vary with total dose? Do we need a kind of a sliding scale that they get bigger as it increases? Can we understand and manipulate mechanisms? If we understood the low dose mechanisms, would we have new targets, for example, for therapy or for radiation protection? And then one of our particular interests is what about other species? Virtually everything in radiation protection at the moment is concerned with cancer induction in humans. But cancer is not a problem in other species, and how do we go about looking at radiation protection of ecosystems, for example, where you might have low chronic doses to multiple species? So I don't need to tell this audience that low dose effects are different. We have adaptive effects, we have hormetic effects, we have homeostatic effects. And what we constantly find, and this has been said a lot here, is that genetic and environmental factors are more important than the actual dose that you give. It's the context and the environment in which you give the dose. So this gives rise to this kind of uncertainty that worries people, because if you have uncertainty and you're a regulator, you're going to regulate for conservative procedures instead of maybe taking a chance. You'll take a chance with animals, but you won't take a chance with humans. So this, I think, underlies a lot of this so-called radiophobia, or extreme protective conservative policies. So I just wanted to put up a cartoon showing the difference between the old and the new paradigms in low-dose radiobiology. The old view was that you hit the cell, you had to hit the cell and deposit energy in that cell. That energy had to cause DNA damage. Pretty much no other target was considered. And that damage, if it wasn't repaired effectively, just transmitted to all the progeny, and they all would carry the same mutation or transformation. But the new paradigm is that you don't even need to hit a cell, that you can sort of create this situation at the system level or the organ level, if you like, and that the result of that is that you get an instability which can give you favorable effects, bad effects, lethal effects. The thing is that you don't know. And therefore the issue for risk is that there's spatial and temporal uncertainty about where the effects are going to happen and what effects they are going to be. And this again feeds into this uncertainty problem. So what we work on most of the time is the bystander effect. This is where you don't need to hit the cell. And as I've been sort of showing this slide over the years, it's got more and more complex. And the most recent addition, there are two recent additions, which is quite interesting. One is UV as being an important signal, and the other is that there is a role, it's been confirmed in 2 or 3 labs in the literature, for exosomes, and we don't know what that role is yet. But the other factors, the old reliables of serotonin is involved, calcium pulses and ion channels, calcium ion channels are involved in the signal production. And then in the response side of the equation, we have, again, the old favorites of reactive oxygen species, TGF-beta, various cytokines, P53, nitric oxide, they're all involved in the response. So it's coming up as a lot of what we've heard already. But the issue again for protection is that a lot of things cause these bystander effects, what we call bystander effects. So how can we define what the unique radiation risk is anymore in this environment where you get bystander effects due to metals, due to various pesticides and other stressors? They can all cause these effects. So what I want to tell you two bits of our recent advances. One is that we have over the years been considering that there could be a physical component to the signal. So there's lots of anecdotal evidence, which I've listed there, which does suggest a physical component. That would be that it's not a chemical transmission. And the one I'm going to, this is a slide of Collins, which is just showing a physical signal, a light signal in this case, that the octopus uses to communicate. So physical light or sound signals are well known in nature. But the experiment that really convinced us that there was a physical signal was this one. We irradiated these zebrafish and we put them in a tank within a tank. So there could be no communication of the signal through the water. Everyone thought that bystander effects were transmitted through liquid medium. But this, clearly there was no way that the signal could transmit to the fish that were in the outer aquarium. But it did. So that meant it had to be light or it had to be sound. We still think sound is involved actually in the fish, but we looked at light first. And any of you that were at the poster session talking to Michelle Lee, this is basically her PhD, was to look at ultraviolet photon emission from cells. And what she did was she exposed cells to tritium, which, as you know, has a very short energy. And then she placed bystander cells in a totally separate flask on top of the tritium irradiated cells. And she was able to get bystander effects in those flasks. So there was no transmission, there was no ionizing radiation, and there was no transmission through medium. And by a lot of various experiments, she was able to show that it was UVA that was doing it. So the issues that we have with this are reconciling it with all the work in the literature that suggests it's a chemical, it's a liquid transmitted chemical signal, determining whether there are other photon energies involved or is it just this UVA, she's collecting UVA photons, determining what the energy transfer vehicles are. Like is there the photon equivalent of an electron transport chain where you get absorption of photons and then emission of lower energy photons, what's going on at the biochemical level? And then determining whether other radiation qualities do the same, because we've only looked at tritium and yttrium-90, which are both beta emitters. This is just showing her procedure. So she has the tritium in the Petri dish and a photomultiplier tube, which is gated to collect UV photons. And her bystander protocol is just shown there. So there's no kind of liquid transmission of the signal. And to confirm that it wasn't sort of stray light or anything like that, she used a UV filter, a UVA filter material, and she was able to cut down completely both the photon emission from the irradiated cells and also the actual bystander effect. And these are just the data again showing the level of bystander effect in the dark bars. And then if you put the filter in, you completely abolish that bystander effect, and they're all the same as the control. And again, correlating the dose of tritium with the photon emission gives you an absolutely near perfect correlation between the clonogenic survival of the bystander cells and the photon emission from the directly irradiated cells. I don't think you could get a more convincing correlation than that. So then she went on to do what we call mix and match experiments. We were trying to pin down what was important for the expression of this UV-mediated bystander effect. So she looked at cells that did or didn't have bystander effects, and she mixed them up so that she put medium from cells that did onto cells that didn't, and vice versa. And her answers, the answers she got, was that the signal is pretty much always produced. Every cell line that we've tested so far can produce the UV photons. And that's, I think, what you'd expect from the physics perspective, that if you have, if you put the energy in and you're exciting presumably the aromatic elements, the sort of pi electron cloud, then you would expect that to happen, irrespective of the genetics of the cell line. But we found a very different answer when we looked at the response. And we found that you had to have p53 functional in order to respond. And if you had p53 null cell lines, you just didn't get a bystander response. You still got the signal, but not the response. And that's something that we think could be really important for radiotherapy, where most of the tumors are actually p53 compromised, but the normal tissue surrounding the tumor would have wild type. So in the future, what we're planning on doing is resolving the medium transfer candidate molecules and the UV work. So we're looking at exosome production in the flasks that receive only the UV signal. And we have actually shown that these cells, the bystander cells, do produce exosomes. So the next thing is to get into exosome profiling. And then we also want to look at other photon energies that might be emitted, and that's planned at the moment. And in terms of understanding the chemistry and biochemistry, well, there's certainly several PhDs, maybe not several lifetimes. So if I have I have 10 minutes. Yeah, 5 minutes. I just want to touch briefly on the other area of research that we're very involved in at the moment. This is where we're trying to look at the controversies. So the low-dose DREF values are still unknown, and the RBE values are still unknown. ICRP use DREF of 2 and an RBE for alpha of 20. But that it's very important to realize that that is just for human cancer. And it's all based on A-bomb survivors, where they extrapolated from high dose to low dose. The role of adaptive hormetic effect is not even considered in radiation protection by the regulators. And as we all know, LNT is the model that's used. So what we think we need to do is look at the low dose region, do the actual experiments, and get the data, and also to re-examine some of the old data in the context of radiation protection at low doses. So I've divided this into what's going on in vivo and what's going on in vitro. I'm summarizing an awful lot of work that we've been doing over the last about 15 years. So in vivo, if you expose fish to chronic low environmentally relevant doses, you get a non-linear uptake. In our case, we looked at radium, and you get a depuration of that radium, or loss of radium, from the fish body over time. And we think that's evidence of adaptation, induction of adaptive mechanisms that get rid of the toxin, in this case radium. And proteomics in vivo from these fish show the induction of a beneficial proteome. So they induce lots of energy mobilizing proteins, lots of scaffold proteins, all the kind of things that you would do if you were trying to kind of overcome a stress. The RBE study in vivo shows minor transient biochemical effects, but the RBE for alpha versus X-rays is 1, so there's no difference. You're not having this big factor of 10 or 20 when you're down in the environmentally relevant dose range. And by environmentally relevant, I mean the levels that are found in the uranium mining areas, like the ponds in those areas. So it's really environmentally relevant. We also found that chronic exposure to gamma or beta in vivo protects against subsequent high dose exposure. So that's kind of all what we've been hearing here, these adaptive hormetic responses. Now, the in vitro data that we have, we've chromosomal instability after acute dose, and we have an alpha-gamma comparison. We have delayed reproductive death or lethal mutations after acute dose, and we have an alpha-gamma comparison. And we have delayed death after chronic exposure, and we have adaptive response data. Sorry. So what's going on in vitro? If you look at single acute doses, you're getting yields of de novo lethal chromosomal aberrations or lethal mutations, and they're constant in the descendants post-acute one-off alpha. So you're getting a consistent, constant effect which is transmitted to all the progeny. And there's no diminution of effect with time out to 120 generations. So I think you can effectively say it's permanent in any biological sense. But there was no difference between alpha and gamma. So again, we're getting an RBE of 1 if you give a single one-off acute dose to cell cultures in vitro. If you look at the effects of chronic doses, previous data that we had showed that a one-off acute relatively high dose of alpha was followed by 400 population doublings by a gamma exposure. And then Xiaopei, our student here, has current experiments where she's looking at chronic exposure to very low dose alpha followed by a one-off low dose gamma. And basically, we're not seeing any signs of these kind of adaptive responses following chronic irradiation in vitro. And if you remember, we did see them in vivo. So we kind of really don't know what's going on. Certainly, it's suggesting that the in vitro models are not realistically reflecting what's going on in the animal models in vivo. And I think that's really important because so much of radiobiology is coming from cell culture work, tumor cell line work, and maybe mouse work as well. But most of the theories have been worked out using in vitro techniques. These are just the data from Xiaopei's direct irradiation showing that after a certain length of time, you do get adaptation in these cells, but when she re-irradiated them, it didn't matter how much chronic dose they'd accumulated. There was no evidence of an adaptive response when they got the high dose. So you're getting an adaptation, an evolutionary adaptation over time to the actual chronic exposure to radium in vitro, but you're not seeing the classic adaptive response which you would expect. You get it after an acute dose, but not after a chronic dose. So my summary and conclusions are that the bystander data suggests that UVA is an important signal which results in exosome secretion from the bystander cells, and that it's probably a protective or homeostatic mechanism. And the chronic versus acute studies suggest that chronic exposure leads to adaptive protective responses in vivo with a low dose RBE of 1, but the in vitro data do not show agreement, and there's no evidence of chronic dose protection against subsequent high dose exposure. And we found that very, very troubling and surprising. And finally, just to be provocative, I can be here, we think that radiation protection in practice doesn't seem to be based on tenable science. So, thank you.
Thank you. Speakers now for questions.
Yeah.
Question just on the difference between in vivo and in vitro. I always think in terms of oxygen, it's the level of the tissue. And radiation effects really pay attention to the local microenvironment oxygen.
No, that's a very good question, because most of our cell culture work, everybody does cell culture with high CO2. So we have an abnormal amount of CO2 to buffer the cell medium. And I think that's probably highly relevant to what's going on.
What I'm saying is that you have hyperoxic cultures as well.
Yeah, there's 20%. Yeah,
right.
yeah.
I don't know what's the in vivo, those same cells, what's the in vivo pressure of oxygen?
It would be way down in the tissue.
Try to do your in vitro work to find more physiologically relevant oxygen.
Yeah, you see, all these cell culture methods have been developed using buffered.
It's a problem.
Yes, but I take your point. We should.
Technologists have learned that the hard way.
Yeah. I know with fish cell lines, you've got to get rid of the CO2 because they just die with CO2. So that might be telling us something too. Yeah.
Just to reinforce, I also agree with the oxygen comment. The
Yeah.
stem cell community has learned that the hard way too.
Yeah.
But the question I had was in the exosomes. Can you speculate what you think might be in there? I know you said you don't know yet, but are you thinking miRNAs?
That's
or
what people are thinking. We haven't, we've only just recently got into the exosome field, and we haven't even started profiling or anything. But the people that are doing that work think it's mRNAs. And that's the hottest area in our field. That's why we got so worried when we had a UV signal, because nobody was listening to us. They were all on the exosome bandwagon. So we decided we had to get in there too. But that's as far as we've got. Yeah.
Have you considered the possibility that when you put the tank within the tank, that the fish are simply watching the other fish and reacting to that?
Like the flies.
Yeah, like some kind of cultural, as opposed to necessarily being transmitted radioactively.
Well, the thing is that there's no visible signs of, like, we don't irradiate our fish to lethal levels or anything like that. They're perfectly healthy. They live out their full lifespan, and all we're seeing is sort of proteomic differences, subtle changes. So I don't know. I mean, yeah.
Just to add in there, we did the experiment as well with aluminium.
Oh yeah, we did.
Oh.
Yeah.
And it still got the same effect.
That was what made us think it might be a sound with our fish. Yeah. Oh. Yeah. Okay.
When you use the epidemiological data for determining the cancer, it already includes the bystander effect. So you don't really need the bystander effect at all to determine the cancer risk from radiation. And low dose radiation shows reduction, high dose radiation shows increase, and there's really no need for the bystander effect.
Yeah, but the epidemiology data is only good down to a certain dose. It doesn't.
It is, if you look at low doses,
Yeah.
the cancer risk goes down. It's good at low doses, and the cancer risk goes down at low doses. And at high doses, it increases. So it's the J-shaped curve. So, I mean, the statement that you need huge numbers to determine the cancer risk at low doses is based on the LNP model
Yeah.
prediction, not
Yeah.
based on actual data. Actual data is it goes down and up. I mean, for example, in Taiwan and many other examples.
Yeah.
So
I
I don't see any reason for bystander effect to come in at all, or to extrapolate the data from high dose to low dose. Because low dose data that shows reduction in cancer.
Yeah, yeah, I was making the point that the regulatory authorities are very worried about these non-targeted effects because I take your point, their underlying premise is wrong. But they are worried about it. They're trying to work out what it means for target theory and, you know, I agree with you. I know what you're saying, but
It already includes the
yeah.
observation includes non-targeted effects, so no need to worry about that.
Okay.
On
Yeah.
the tank within the tank experiment,
Yeah.
where you speculate that the UVA is transmission, signal transmission medium, have you looked into the UVA transmission properties of glass cage and of the liquid medium?
We have, we did it in a sort of a weird way. We measured, using the photomultiplier tube, we collected the photons coming through the glass. And then we put this filter material in to block the UV signal, and the effect went away. So, okay. Okay,
Okay, thank you.
thank you.